Hello everyone. In my previous two videos, I discussed in detail about the concept of five kingdom system of classification proposed by R.H. Whittaker in 1969. In my last video, I also focus on Archaebacteria, which is the type of organism which he placed in Kingdom-Monera. Now in this video lecture, I am going to focus on Eubacteria. The modern day bacteria which generally present everywhere, that means they are omnipresent and they play a very important role in number of different type of industries. Now in this video lecture I am going to discuss about cell structure of the eubacteria, then size and shape, gram character, reproduction that occur in the eubacteria, nutrition and regarding their motility. Okay, so first is cell structure. Here you can see eubacterial cell. Generally bacteria are prokaryotes, so they do not have well developed nuclear membrane, nucleolus, nucleoplasm and histone protein. DNA is directly suspended in the cytoplasm and it is called as nucleoid. So bacterial DNA do not have separate compartment which is nucleus like eukaryotic cell, but it generally present directly suspended in the cytoplasm. So we call it as a nucleoid. Then cytoplasm of the prokaryotic cell also show presence of inclusion bodies. These are the storage house of the bacterial cell which are responsible for storing food material. Then cytoplasm of the bacterial cell also have ribosomes which are 70S type. We know that prokaryotic cell always have protein synthesis organ, that is ribosome 70S type, which divided into two subunit. Large subunit is 50S subunit while small subunit is 30S subunit. Okay, then cytoplasm of the prokaryotic cell do not show any type of a cell organelle like mitochondria, chloroplast, endoplasmic reticulum, Golgi complex. All advanced cell bound organelle which is present in the eukaryotic cell is absent in prokaryotic cell. So when we compare this cell with the eukaryotic cell, we can say that it is a very simple cell. Okay, now prokaryotic cell on the surface show thin hair like long structure called flagella. This flagella help bacteria at the time of motility. So the organ which is responsible for locomotion in the bacteria is flagella. Flagella are less in number when we compare with another small hair-like structures which is present on the bacterial cell, and they are pili. So function of the pili is completely different from the flagella. Generally, pili are made up of a protein called pilin and they help in attachment of the bacterial cell to the solid surface. So pili function in attachment as well as in conjugation. Okay, conjugation is the process in which genetic material is exchanged between two different cells. Okay, and for conjugation, two cells must be get attached with each other, and that attachment is generally carried out with the help of pili. Then bacterial cell also show presence of a polysaccharide glycocalyx layer around the cell, which is called as capsule. These capsule are not present in every bacteria, okay, but some type of bacteria generally show presence of capsule. There are two different type of capsules which is present on the bacterial surface, like homopolysaccharide capsule and heteropolysaccharide capsule. Streptococcus mutant is the type of a pathogen which is responsible for tooth decay, and the bacteria have a homopolysaccharide capsule, while Diplococcus pneumoniae, which is the pathogen responsible for pneumonia, have heteropolysaccharide capsules. This capsule which is present on the bacterial cell play a number of different type of functions, like they help in the attachment to the solid surface. Then they also prevent phagocytosis. That means when bacteria have this capsule, it can be protect the bacteria from the attack of the immune system inside the host. Now this capsule also protect the bacteria from bacteriophages or viruses which kill bacteria. So these capsules are responsible for number of different type of important functions inside the bacteria. That means they help bacteria to survive in unfavorable conditions. Sometimes this capsule attached to the water molecules and they prevent dehydration. Okay, so at the time of dehydration, this capsule have ability to protect the bacterial cell. This is all about the structural aspects of the eubacteria. Now I am going to focus on size and shape. Generally, the detailed study of the prokaryotic cell is become possible only after the discovery of electron microscope after 1914. Okay, so after discovery of an electron microscope, scientists are able to study the each and every organ of the bacterial cell separately, and it clearly demonstrate that the bacterial cell have different type of shapes. Like some bacteria have circular shape which is called as cocci. Okay, then sometime two cocci remain attached to one another and it is called as diplococci. This happened at the time of reproduction when bacteria divide in a single plane, and after division two cells remain attached with each other. It is called as diplococcus. And if bacterial cell remain attached for multiple divisions which occur in the single plane, then this type of a chain of the cocci is produced, which is called as streptococci. Okay, so round or circular bacteria generally present in cocci, diplococci or streptococcus forms. Then bacteria also show cylindrical elongated rod-like structure called bacilli. Bacilli. Sometimes bacteria is present in spiral conformation which is called as spiralum. Okay, and comma-shaped bacterial cells are also present. They are called as vibrio. So all these are the different shapes of the bacterial cell. Generally, size of the bacterial cell vary bacteria to bacteria. For example, cocci show diameter of 0.25 to 1.25 micrometers. Okay, while bacteria like bacillus show length from 1 to 3 micrometers. So we have discussed cell structure and size and shape of the prokaryotic eubacteria. Now I'm going to focus on gram character. Hence, discover a technique of staining bacterial cell, and on the basis of the result, he categorized the bacteria into two different groups like gram positive organism and gram negative organism. What is the major difference between gram positive and gram negative organisms? The difference lies in the cell wall structure of the bacteria. If I enlarge this portion of the bacterial cell wall, it will going to look like this. If it is a gram positive bacteria, then it have cell membrane which is made up of phospholipid bilayer. The cell membrane have hydrophobic tail facing inward and hydrophilic head facing outward. Okay, this is the fluid mosaic model of the cell membrane which show phospholipid bilayer and different type of protein embedded in it. So this is the structure of gram positive bacterial cell membrane, and outside the cell membrane a thick peptidoglycan layer is present called cell wall. So gram positive bacteria show thick peptidoglycan layer outside the cell membrane, and this peptidoglycan layer also show strands of protein called teichoic acid. Okay, now this peptidoglycan layer is a rigid structure which prevent the entry of number of different type of toxic molecule inside the cell, as well as this peptidoglycan layer is responsible for maintaining cell structure and shape at the time of different environmental conditions. This is all about the gram positive bacterial cell wall. When we compare this with the gram negative organism, you can easily find the difference. In case of gram negative organism, cell membrane remains same, which is a phospholipid bilayer, but outside the cell membrane, you can find peptidoglycan layer, which is very thin when we compare with a gram positive bacterial cell wall. So in case of gram negative organisms, peptidoglycan percent is very less, and outside the peptidoglycan layer, outer membrane is present. This outer membrane is again made up of lipopolysaccharides, and this outer membrane show number of different type of openings for transport of the molecule, which is made up of protein called porins. Okay, so this outer membrane is absent in the gram-positive organisms. The fragments of the outer membrane of gram-negative organisms serve as endotoxins. This is responsible for fever, as well as septic shock induction in the host organisms. So when we compare positive and gram-negative organism, we can find the difference, like in gram-positive organism, peptidoglycan is very high or thick layer of peptidoglycan is present, but in case of gram-negative organism, thin layer of peptidoglycan is present, and outside the peptidoglycan, outer membrane is present. And on the basis of this cell wall difference, organisms like Eubacteria are divided into two types, gram-positive and gram-negative. Okay, at the time of gram staining, crystal violet is retained by this gram-negative or gram-positive organisms, so they appear violet, while saponin, which is a secondary strain, is going to stain gram-negative organism in different color. Okay, I'm going to discuss about gram staining in my upcoming videos. The next point is reproduction. Bacteria show two different type of reproduction. Mainly bacteria are reproduced by asexual reproduction, but sometimes sexual reproduction is also observed. Asexual reproduction in the bacteria occur by different method like binary fission, budding, fragmentation. In this figure I have shown, first is binary fission and second is budding. So both these are the type of reproduction which occur inside the bacterial cell, and this is the asexual mode of reproduction. In binary fission, a normal bacterial cell undergo DNA replication, and by the process of DNA replication, two copies of DNA are produced, and then by cytokinesis, division of the cytoplasm occur by niche formation, and from single cell, two different cells are produced. Here you can see when DNA gets replicated, two copies of DNA are produced, and then each copy enter in a separate daughter cell. So this is binary fission. Now in case of budding, instead of cytokinesis, a simple bud is produced after DNA replication on the bacterial cell. One copy of genetic material is enter in the bud. The bud enlarge in the size in a stepwise manner, and at the end this bud detach from the parent cell, and now this bud form new bacterial cell. This is called as budding. Another type of asexual reproduction is fragmentation. Now sexual reproduction is not common in the bacteria till they have reported three different type of sexual reproduction in the bacterial cell, like transformation, transduction and conjugation. What is transformation? Transformation is transfer of genetic material like DNA inside the bacterial cell right from the environment. That means bacterial cell become competent, and then this competent cell directly receive genetic material from the physical environments without making contact with the donor cell. So in case of transformation, directly DNA enter into the bacterial cell, and this phenomenon is discovered by Griffith in 1928. Which is we discussed in detail in my previous video entitled Discovery of Genetic Material. Okay, so transformation is the physical transfer of DNA from one cell to another cell without making a direct contact. Okay, now transduction is the process of transfer of the genetic material from donor cell to recipient cell with the help of bacteriophage. Bacteriophages are the viruses which having ability to infect the bacteria, and these viruses are sometimes responsible for transferring genetic material from one cell to another cell. This is the transduction. While conjugation, name itself suggests the process in which two bacterial cells come close to one another, then they form a physical contact, and by physical contact, the genetic material from one cell enter into the another cell. So this is called as conjugation, and in conjugation process, these pili help. Okay. Now, these three methods are not actually the processes in which number of cell increases. So they are not like asexual reproduction, but as genetic material transfer from one cell to another cell, we can consider these three as method of sexual reproduction, because two different cells are involved and genetic material get exchanged. Now the next point is nutrition. Generally bacteria are heterotrophic, that means they are unable to make their own food material. They require help of other organisms. Okay, so we call it as a heterotrophic, but sometimes some bacteria are autotrophic, able to produce their own food material with the help of photosynthesis, like cyanobacteria, because they having light absorbing pigment like chlorophyll. Some bacteria are chemosynthetic, that means they are going to use sulfur as a terminal electron acceptor, and the process in which they are able to prepare their own food material. The process is quite different from normal photosynthetic process. So there are two different mode of reproduction which is observed in the nutrition which is observed in the eubacteria. They are autotrophic and they are heterotrophic. Now the last point is motility. Some bacteria show ability to change the position, that means they have ability to show movement, and this is happened because they have this cell hair-like structure called flagella. Flagella is the organ which is present attached to the cell wall of the bacteria, and that flagella help in the locomotion or the movement of the bacteria. Generally, bacteria show different type of arrangements of the flagella on the cells. Like sometime single flagella is attached to the one end of the bacterial cell, and this type of flagellar arrangement is called monotrichous arrangement. Okay, there are four different type of arrangements, out of which first is a monotrichous, in which single flagella is present at one end of the bacterial cell. Sometime many flagellas or a bunch or a group of a flagella is present on a single end of the bacterial cell, which is called as lophotrichous. Okay, second type of a flagellar arrangement. Now in third case, many flagella are present all over the bacterial cell surface, and it is called as peritrichous. While in case of last arrangement, two flagella are present on both the end of the bacterial cell, it is called as amphitrichous. So these figures explain four different type of flagellar arrangement which is present on the bacterial cell, like monotrichous, single flagella at one end, lophotrichous, group of flagella at one end, peritrichous means number of flagella evenly distributed on the bacterial surface, while amphitrichous means two flagella at two different ends of the bacterial cell. Now in last part of this video, I am going to discuss about the structure of this organ, flagella, which is present on the bacterial cell. Generally, under the electron microscope, flagella is an organ which is divided into three different parts, like filaments, hook, and basal body. Filament is a thin hair-like structure which is present, and the filament is mainly made up of protein flagellins. Okay, so single protein subunit is flagellin, and many such subunits come together to form long chains of the protein, which is called as filaments. Now this filament is actually responsible for driving force or movement of the bacteria. Now this filament is attached to hook. So this hook is the second part of the flagella which help in binding filament with the basal body. Now hook is attached to the central rod, and this rod is inserted into the cell wall and cell membrane of the bacteria. If it is a gram-negative cell wall and cell membrane, then you can see four different type of rings attached to the rod of the flagella. These four rings are L ring, P ring, S ring, and M ring. Out of the four rings, L ring is present in the outer membrane. Okay, this is the outer membrane of the gram-negative bacteria, and L ring of the flagella is present in the outer membrane. L for lipopolysaccharide. We know that the outer membrane of the bacteria is made up of lipopolysaccharide. Then second ring which is present in the bacteria is present in the peptidoglycan layer, which is the cell wall, and now this ring is called P ring, P for peptidoglycan. Now third and fourth ring is present in the cell membrane of the bacteria, out of which the superficial ring is called S ring, S for superficial, while last ring or the fourth ring is M ring, M for membrane. Okay, so two rings are present in the cell membrane. So when we study the structure of flagella in gram-negative bacteria, we can see four different rings at four different positions in the outer membrane, peptidoglycan layer and cell membrane. When we consider flagella of gram-positive bacteria, only two different rings are present, one in peptidoglycan layer and one in the cell membrane. This basal body is responsible for rotating the flagella in clockwise as well as anti-clockwise, and due to this rotation, energy is used and bacteria is moved from one side to another side. So flagella play important role in locomotion in eubacteria. Generally, bacteria use this flagella for movement, and the movement is called taxis. When we consider chemotaxis, it is the process in which bacteria move from one side to another side by chemical signal, like from site of low nutrient content to high nutrient content site, bacteria can travel. Okay, it is called as chemotaxis. Sometimes bacteria move from region of high toxic concentration, toxin concentration to low toxin concentration with the help of flagella, and the process called chemotaxis. Sometimes aerotoxic is also, or oxytoxin or taxis is also occur, in which bacteria move from region of low oxygen concentration to favorable oxygen concentration region, which is called as oxytaxis. And another concept is there called phototaxis, in which bacteria move from region of low light intensity to high light intensity or optimum light intensity, where bacteria can perform photosynthesis and prepare their own food material. So in this lecture, I discuss in detail about the cell structure of the bacteria. Cell of the bacteria show different type of part or the structures like flagella, pili, capsule, cell wall, cell membrane, cytoplasm containing DNA in the form of nucleoid, mesosomes, 70S ribosomes, inclusion bodies. Then I discuss about the different shapes of the bacteria, which include circular bacteria, which are cocci, then two cocci attached to each other called Diplococcus, Streptococcus, Bacillus, Spirillum and Vibrio. Then we discuss about the gram character. Hans Crabbe distinguished bacteria into two different types like gram positive and gram negative. Gram positive organism have cell membrane and thick peptidoglycan layer. Cell membrane is a semi-permeable membrane which is responsible for maintaining the traffic in and out of the cell. Then gram-negative bacteria show cell membrane, thin peptidoglycan layer and outer membrane, which is made up of lipopolysaccharide. Then we discuss about the reproduction. Bacteria show two different type of reproduction, asexual and sexual reproduction. Example of asexual reproduction is binary fission and budding, while the example of sexual reproduction in the bacteria is transformation, transduction and conjugation. Then we discuss about the nutrition. Bacteria show two mode of nutrition, autotrophic as well as heterotrophic mode. And last we discuss about the motility. Motility of the bacteria is due to special organ called flagella. Generally four different type of arrangements of flagella are present, like monotrichous, lopotrichous, peritrichous and amphitrichous. And finally at the end of this lecture we discuss about the structure of the flagella, which having three different parts, mainly filaments, hook and basal body. In this lecture I discuss eubacteria in detail. Now in my next video I am going to focus on last type of organisms which are present in the Kingdom-Monera. They are mycoplasma. Thank you.